Hello friends, Janvi here. Welcome to my YouTube channel. Today I have come up with another topic to discuss with you. It will be about genetics and again it will be made into a multiple videos. So please look forward for more videos. Before we start today's video, if you like my channel and videos, please subscribe to it and share it with others. Hello friends, today we are going to talk about a very important topic of biology called as genetics. So today I am going to cover few important concepts regarding the genetics. They are definition of genetics, common terminologies used in genetics, Mendelian work, mainly monohybrid cross and dihybrid cross, laws of heredity, test cross and back cross, incomplete dominance, and the structure of the DNA, because DNA is the genetic material which passes the information from one generation to another. Now we are going to talk about the genetics and common terminologies used in genetics. First, let me tell you what is genetics. Genetic is nothing but the mechanism which deals with the heredity and causes of variation in living being. To understand the genetics in clear cut, we have to know certain terminologies which are used in genetics. The common terminologies which are used in genetics are allele, autosome, allosomes, filial generation, and the gene. What is an allele? Allele is nothing but one of the two or more forms that can exist at a single gene focus, distinguished by their differing effects on the phenotype. Example, hair color. What are autosomes and allosomes? Autosomes are the chromosomes which are not associated with the sex, whereas allosomes are the ones which are associated with the sex. So let me explain you, when we talk about Mendelian genetics, we come across two main things called as F1 generation and F2 generation. The meaning of F is nothing but the filial generation, the generation of progeny controlled in a series of crosses starting from the parent. Next, the basic fundamental concept of genetics called as a gene. What is a gene? The gene is nothing but the physical and functional unit of heredity which carries the information from one generation to another. In a group, we will call gene as an DNA. Now let's talk about the Mendelian work. The Mendel is the person who actually studied how the genetic information will pass on from one generation to another. So actually he did some experiments using the pea plants which showed how the genetic information is passed from one generation to another. Mainly in this slide we are going to explain about a monohybrid cross and a dihybrid cross, showing how the characters are passed from one generation to another. And very important is why he chose the pea plants for all his experiments. Let's talk about the monohybrid cross. The word mono says we are taking only one character at a time to explain the cross. So the Mendel also took only one character at a time. Mainly, I am taking an example of the height of the pea plant to explain the same. So the height is a character. When we talk about the height, two things will come into your mind. One is tall and one is dwarf. So taking these two things at a time, we are going to explain the monohybrid cross. Before we go into the monohybrid cross and the dihybrid cross, we have to see why Mendel selected the pea plants for his experiment. As I have said, the pea plants have some history, so that he selected the pea plants for his experiment. The why he selected the pea plants, the reasons are here. One is variation, second one reproduction, third one controlled mating, fourth one short life cycle, fifth one large number of offsprings, fifth one convenient in handling. These features have helped the Mendel to select the pea plant for his experiments. So, if we start with explaining the each character, like controlled mating, that means what? The crossing over or hybridization techniques are very easy to handle with the pea plants. And the life cycle, short life cycle, whatever the plants we will select for an experiment, it should have a very short life cycle, so we can easily study the changes or variations which will take place in those plants, and at the end we can conclude what are the things we have obtained from these plants because of their short life cycle. And large number of offsprings. What do you mean by large number of offsprings? That means while doing an experiment, we will get more than 2 or 3 offsprings at a time. Or it may be for in the monohybrid cross, we are getting 4 uh, plants at a time. That means what? There are large number of offsprings in these cases. And last one is convenient in handling. What is the meaning of convenient in handling? It is very easy to handle it. Whereas other plants, it may be difficult to grow it or hybridize it. But these pea plants are very easy to hybridize. And the variation means what? The changes which can take place, is the change we can recognize those changes or not? Is it at the physical level or it is at the genomic level? Keeping all this in mind, we have to select the plant. So keeping all these things in mind, the Mendel selected the pea plants for his experiments. Okay, now we will discuss with the monohybrid cross. So to start with the monohybrid cross, I am taking an example of the height of the pea plant. So just we have to discuss it using a diagram. It is very simple to understand the monohybrid cross. Here you can see in a diagram, I have taken the tall plant and I have crossed it with a dwarf plant. So gametes here are the big T and the small t. So when we cross it with each other, I will get the heterozygous tall plants in the F1 generation. Now we will see what characters will appear in the F2 generation. So to start with the F1 generation, I am crossing the F1 parents, that is the heterozygous tall plant with another heterozygous tall plant. The gametes here are the big T and the small t of one parent of heterozygous tall and big T and small t of heterozygous tall of another plant. So when we cross it with one another, we can see in this diagram the big T of the one parent is crossed with the big T of another parent to get homozygous tall plants. And big T of one parent is crossed with the small t of another parent to get an heterozygous tall plant. And the same cross is done with another parent, taking the tall plant of one parent and crossed with the dwarf plant of another parent, we will get again a heterozygous tall condition. Whereas the dwarf T and the small t will combine together to form a homozygous dwarf plant. So totally in the F2 generation, we will get 1:2:1 ratio. That means homozygous tall plant will be 1, heterozygous tall plant will be 2, and homozygous dwarf plant will be 1, and this will be appearing as a genotypic ratio. The phenotypic ratio of the plant is 3:1. That means Three tall plants are appeared in the F2 generation with one dwarf plant. That means to see the plant, we will distinguish either it is a tall or it is a dwarf. We will not be able to understand whether it is a homozygous or a heterozygous in this condition. So phenotypic ratio is also important when compared to the genotypic ratio. The test cross. The cross which is done between the F1 generation and the recessive parent is called a test cross. The cross which is done between the F1 generation and dominant character of the parent is called as the back cross. Let's talk about the back cross. In back cross, we are taking an F1 generation heterozygous tall plants and will cross it with an parent of the dominant character. Here the dominant character is the tallness. So in a back cross, we are crossing the F1 generation with the dominant character of the parent. So whatever we will get in the back cross is nothing but the homozygous tall condition and a heterozygous tall condition. 50% of the plants will be homozygous tall, whereas 50% of the other plants will be heterozygous tall. So let's talk about the test cross. What is a test cross? It's very simple. We are taking an F1 generation tall plants that are nothing but the heterozygous tall plants, and we are crossing with the recessive parent, that is nothing but the gene which are recessive in expressing their character. So whatever we'll get in F2 generation are 50% tall plants with a heterozygous condition, whereas another 50% are homozygous with a dwarf character. This is called as test cross. The test cross are nothing but the cross which is taken between the F1 generation and crossed with an recessive parent. And whatever the progeny we will get, that progeny will have 50% of the heterozygous tall plants and the another 50% is of homozygous dwarf plants in them. Today I am now talking about the laws of heredity. The two laws of heredity is as follows. They are law of segregation and law of independent assortment. Now let me explain about law of segregation. It is also known as law of purity of gametes. The law states that the hybrids or the heterozygotes of F1 generation have two contrasting characters. One is dominant and another one is recessive. These alleles do remain together for a longer time but do not contaminate or mix with one another, and they will separate or segregate at the time of gametogenesis, so that each gamete receives only one allele of character, either it may be a dominant or recessive. To explain the law of segregation, the best example is a monohybrid cross. As I have explained you before, what is a monohybrid cross, we can easily know how the law of segregation work. That means the tall character and the dwarf character does not mix with each other, but they will pass on to the next generation without mixing. That means which gene is dominant, that gene will appear in an F1 generation. So the gametes will always segregate or separate at the time of gametogenesis. Now we will talk about the second law of heredity called as law of independent assortment. When two pairs or characters of the traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of the character. Such a ratio was observed for several pair of characters by Mendel. To explain the law of independent assortment, I am taking an example of a dihybrid cross in which you will come to know how the segregation or the characters are separated and they are independent of one another. Hence, we will call both the laws of heredity as a very important concept of genetics, and there are certain deviations from this law which we will study in the further slides. Now we will see the fast recap of what we have studied in this slide. First, we have learnt about what is the definition of genetics, the common terminologies which are used in genetics. Third thing is a monohybrid cross and why the Mendel chose pea plants for his selection. And fourth one is a loss of heredity. And in the next video, we are going to learn about dihybrid cross, incomplete dominance, and other things about the deviations in the Mendelian law.